Good morning and welcome to the Ohio State University Center for Clinical and Translational Science 8th Annual Scientific Meeting. My name's Dr. Rebecca Jackson. I'm the director of the OSU CCTS. On behalf of our partners, Nationwide Children's Hospital, the Ohio State University Wexner Medical Center, the Office of Research for the Ohio State University, the multiple colleges across our university, our stakeholders in the communities, and the CTSA Consortium, I'm really excited to have the opportunity to spend the next 3 Tuesday mornings together with you, really exploring opportunities for us to learn from an unexpected and certainly challenging event that occurred over the last 12 months, the COVID pandemic, as a case study for ways to pivot, yet keep our true vision for improving translational science moving forward. Next slide. It has been almost 160 years since the US government passed the Morrill Act, which actually created land-grant universities to create improved opportunities for education and training throughout the United States. The Ohio State University and its partners are a land-grant university and took that mission very seriously. Approximately 40 to 50 years ago, however, rather than simply being a disseminator of knowledge, the university made a persistent and strategic decision, in fact, to generate new discoveries that were going to translate to improvement, not only for our state, our nation, but globally as a sacred social compact with the communities that we serve. We brought together our basic science discovery, our clinical research, our community efforts, and our policy to work together in new ways to think about addressing some of the greatest problems that we're facing us today. Next slide. The guiding principles of the CCTS, which actually has as its central core to enhance the translation of today's discoveries to improve health, are really about the people who move that science forward. All of you who are attending this program today and are involved in so many incredible scientific opportunities here in collaboration with other institutions, and in fact, in collaboration with our communities, our state, and our nation. As Woody Hayes once said, you win with people. And that's making sure that we actually include all voices at the table in order to be able to do research that has the greatest impact. What drives that forward then is an approach towards stimulating innovation and experimentation. And we really fell on these core guiding principles as we addressed the COVID-19 pandemic and some of the pivots that were required in order to continue to meet our central mission. The CCTS does this through really 4 primary approaches: to convene groups of individuals together, to create communities of practice, and to enhance collaboration through connections and other opportunities to begin to bring multiple perspectives to actually do not only multidisciplinary research, but in fact to move into that magic of transdisciplinary research. We catalyze new research by putting forward funding opportunities and creating an enabling environment by bringing together multiple types of resources that actually will facilitate the work of our scientific community. And then we use data-driven decision-making, actually understanding what is the impact of the programs that we have. Have we made life easier for you? Are we making things more efficient, safer, and of higher quality in order to impact the science that is so critical to our central mission? Next slide. We do this by creating a foundation of resources. Certainly critical within this is informatics and new IT approaches to take data to knowledge and to learn from the vast amount of not only patient data, but basic science data, community-based data, public health data, and policy, and to begin to bring these into data ecosystems to ask questions that we could never answer before. We bring together scientific cores across the entire translational spectrum, from basic science discovery to the bench, to the bedside, to the community, and work those together so that there's not the impediments or the barriers to being able to move forward your work. We provide educational programs, tools, and other resources so that we have a well-prepared educational workforce, not only of investigators, but in fact of the entire translational research team, which includes our stakeholders and community members in authentic partnerships. We do this then through kind of three primary pillars: to train and cultivate a translational science workforce, to enhance scientific innovation, to really catalyze the trajectories by providing the resources that are necessary, and then to think about new ways to innovate research processes that can improve the quality, safety, and efficiency. And that by bringing together this strong foundation in collaboration with other interdisciplinary centers like the Comprehensive Cancer Center, the Infectious Disease Institute, the Office of Research Cores, and others, our goal is to really have a translational research engine that propels us to the ultimate objective of an environment that actually embodies sustainable and innovative clinical and translational science discovery and implementation. Next slide. Our vision really has been unchanged over the 15 years that the OSU Center for Clinical and Translational Science has been funded. That we focus on creating a healthier Ohio, a nation, and a world. And we continue to look at not only those major health issues across, again, the entire translational spectrum, as well as emerging issues. Next slide. And of course, nowhere were we prepared for the changes that have occurred in 2020, when the global pandemic of COVID-19 had a significant impact on every community, on every individual, and in fact, on the entire translational science research infrastructure. But as scientists, as committed individuals, as a community, we felt it was critical that we continue to move forward our mission of advancing discovery to improve health. And we simply broadened that base. So rather than changing our approach, we actually protected that vision of things and looked at ways, how we continue to make advances? Where were our opportunities to pivot? How could we take the great strengths across Nationwide Children's Hospital, the Ohio State University, our collaborations with other universities and the CTSA consortium, and actually affect the COVID-19 pandemic? So I'm going to give you a couple of examples of the way that we actually actualize this pivot rather than pause. Next slide. The first of these is that we needed to balance the things that were capable of being done within the social distancing and some of the early stages during the early surge, and then the later surge, to actually meet the community scientific needs. There was clearly still a great need to catalyze science, to bring together multidisciplinary and interdisciplinary teams across divisions, departments, colleges, and universities to be able not only to address COVID-19, but to continue to move forward our efforts broadly about improving the health of our nation. And so we pivoted our pilot program in the last year to actually focus early on those kind of studies we can do when laboratories were closed. And so we funded 21 new scientific teams that received pilot funding, focusing on secondary data use, as well as developing out new approaches and applications of artificial intelligence, machine learning, and deep learning to get insights out of the large amount of data that was present. We actually continued to drive forward science. And so when our cores were closed, we pivoted some of our vouchers to be able to address other things that were necessary to continue to make advances that were able to be done when laboratories were closed. And so since 2009, we have given more than 259 vouchers, more than 90 in our grant year 3, which extends through the end of May 31st. So please think about applying for these. And we developed a new program with a higher amount of voucher amount early on to actually catalyze research that was focused on COVID-19 activities and specific types of utilization of cores that were necessary, again, across the large translational science spectrum. We continue to have consults and expertise available, but we pivoted our strategies, for example, in our recruitment core to developing out even stronger toolkits that allowed us to use digital strategies and inform research teams about how they can work within the new environment in order to safely reach out and engage individuals. And with all of the creativity, innovation, there was also a much greater emphasis on inventions and dissemination. And so we took the inventor workshops and began to highlight some of the great advantages and some of the great accomplishments of research teams across Ohio State and nationwide, and in creating solutions for some of the greatest problems associated with COVID-19. Our educational programs expanded, but now in a virtual environment, and more than 2,100 individuals attended more than one CCTS educational program. And new resources that you'll hear about soon include COVID-19 data registries, including the involvement in the National Collaborative COVID Network, or N3C, and the creation with a community of practice of more than 150 individuals actually advising it of an OSU COVID data mark for being able to actually do discovery and testing. Next slide. There is also great insight and new methods that developed. And you're going to hear over the course of today, next week, and the following week about some of these new methods in research and education that have actually been developed within the challenges of COVID-19 to really address some of the barriers that in fact are likely to have great value to be adopted as best practices in moving forward. Next slide. So today, our focus is on research pivots and rapid response and understanding for a number of different case studies, the best practices that can be learned. Understand that this is not a COVID-19 conference, but it's really asking you to think differently, to go beyond the study and to think about what are the lessons that were learned. How, by implementing new strategies and rapid responses, did we find new ways of doing things that in fact should be adopted and translated to the future in our near normal? And what opportunities are there for us to leverage this large ecosystem at The Ohio State University and Nationwide Children of cores, resources, people, and other that can support that ability? Hopefully there will never be a pandemic again exactly like this, but there are certainly always challenges that occur within research. And if we learn ways that we can adapt and do a better job, these are critical lessons learned to move forward. Next slide. Lastly, it is important to remember that this is really about you. The CCTS, The Ohio State University, and Nationwide Children's Hospital are really here to support your scientific success. Because in fact, clinical and translational science is not individual science. To be able to translate discovery to actually changes in the way that we deliver health to create a healthier state, nation, and world is team science that requires multiple perspectives, multiple expertise, and individuals from multiple backgrounds, from scientists to our trainees, to community members, to our entire research teams. I think Henry Ford probably said this better than anyone. Coming together is a beginning. And certainly we did this in the early responses to the COVID-19 pandemic. Keeping together is progress. So how do we continue to learn from these lessons and move them forward? And critically, our opportunity and our success in the future will be by working together, which is success. Thank you all for attending. I hope this is an important and eye-opening opportunity. And of course, go Bucks. Next year starts today. And we all have the opportunity to learn from what we've learned in the field. I'm going to turn this over to our first speaker, Dr. David Boulware. We are thrilled to have him join us here at our annual scientific meeting, who is going to talk about lessons from virtual trials in a time of a pandemic. Dr. Boulware, welcome.
Great, thank you, Dr. Jackson. It's great to be here. Thank you for the invitation to speak. I'm Professor David Boulware. I'm a infectious disease doctor in Minnesota, also at land-grant university. And I'm going to talk less about the trial results, but a little bit more about how we actually did these trials. So lessons from virtual trials in a time of pandemic. And so I don't have any relevant disclosures. And so we conducted a series of randomized controlled trials on hydroxychloroquine early in the pandemic back in March and April. And so we conducted 3 trials, the first of which was published in June. The second was published in July. The first was on post-exposure prophylaxis of people exposed and trying to prevent infection. The second one is on early treatment. And the third was on pre-exposure prophylaxis for healthcare workers, or PrEP. And we conducted these sort of rapidly on a fairly limited budget of probably was about maybe $200,000 in total to run 3 nationwide RCTs. And so I want to talk a little bit about just the story behind this, and hopefully as some lessons for you. And so what did we learn, and how did this come about? Well, first off, most of my research, for people who actually know me, I'm a cryptococcal meningitis researcher. So I do fungal clinical trials in Africa and work primarily in Uganda, work a little bit in South Africa, and I've worked in Ethiopia as well. And most of my work is all investigator-initiated clinical trials. And so this is a neglected disease, and so there aren't big pharmaceutical companies lining up to really help support this. And so we know how to do clinical trials. And we were supposed to attend a HIV conference that was held in Boston in early March last year. And this was right around the time when community transmission was beginning to happen. And the conference was canceled at the last minute. So on the Friday, I think it was Friday evening, it was canceled for the following Monday. And so my team, basically, all of us were going to go, and we were presenting abstracts on tuberculosis, on cryptococcal meningitis. We had like 4 days of free time. And so that never happens, like 4 days of complete, nothing on my agenda. And so I came back from NIH study section and said, Monday morning, let's meet at 9 a.m. and we're going to do a clinical trial. And we sort of rapidly pivoted on March 9th to begin this. And so there's a number of sort of junior investigators, and we have sort of ID docs, and we've got pharmacologists and biostatisticians on our team. And so we basically, pitched in all together and worked on this. And so the timeline sort of was pretty rapid. And so we started the idea on March 9th. And we started our trial on March 17th. And so within that 8 days, we wrote a protocol, we designed our CRFs, built a REDCap database, sent off an FDA investigational new drug application to FDA, got IRB approval, Like all that happened in 8 days, which is like an act of God. It's in retrospect, I'm not sure exactly how we did it, but we knew what we needed to do. We ordered the drug, we got a pharmacy plan, we got the randomization schedule, and figured out how we're going to do this. And we started on March 17th, and thereafter things sort of were off to the races. And we initially started with post-exposure prophylaxis, and we focused on that because no one was really focused on outpatient illness. And so there were a lot of hospital-based, trials that were ongoing and people talking about stuff, but no one was talking about outpatients and thinking about trying to interrupt transmission. We thought, well, what about, post-exposure prophylaxis? And once we started enrolling people, we realized a lot of people were already symptomatic. And so we should also do early treatment. And so on March 22nd, we opened sort of a second trial with a protocol amendment to basically also enroll people within the first 4 days of their symptoms. And then we posted on Twitter about this and sort of, social media dissemination, and some people from Canada said, hey, can we participate? And we're like, sure, why not? And so we gave them the protocol, gave them our REDCap database, and they basically replicated the study in Canada. And so we went from, that opened up in Canada on sort of March 24th. And so in rapid succession, we basically went from an idea to a clinical trial. And this was based on an internet-based trial. The trial ran for about 6 weeks and was stopped on May 3rd. And then we rapidly, wrote up the results and submitted to a journal by May 6th for both of the two trials. And then the third trial, the PrEP trial, pre-exposure prophylaxis ran for 3 months, and that finished in early July and was submitted for publication rapidly thereafter. And so this timeline was sort of compressed, but we knew what we needed to do, and we had a really good team that had worked together before. And so the trial itself is a pretty standardized, it was a randomized trial. We're looking for a reduction in illness, and for our early treatment, we were looking for a reduction in hospitalization. And we assumed sort of a 10% rate of infection in our post-exposure prophylaxis trial. And we assumed about a 20% rate of loss to follow-up. And thinking about an internet based virtual trial, we've never done this before. I guess no one really had done this before. And so we didn't know what the loss to follow-up rate was going to be for a pharmaceutical, drug intervention trial. So it turned out that our rate of illness was a little bit higher, and our rate of loss to follow-up was under 10%, which was pretty good. We were pretty happy with that. And so our ultimate sample size we needed was a little bit lower, which was good. And overall, sort of the process of how this worked is we used a REDCap database, and basically people self-screened themselves, consented, and then we shipped out the medicine. We had a follow-up survey the following day to verify that they received the medicine, and 98% of people did. FedEx lost some packages and stuff, and so sometimes we had to track stuff down. And then we did some follow-up surveys on day 3, day 5, day 10, and day 14, where these were automated surveys that went out. I'm going to talk a little bit more about this in a bit. And so how do we screen people? And so this was, I thought, was self-evident, but I guess it's a little bit creative. So we used a two-part sort of enrollment thing. And the first was a screening, which basically assessed the inclusion and exclusion criteria for the trial. We didn't publicly post these like, hey, these are the exclusion criteria. They were on clinicaltrials.gov, but we didn't tell people exactly what the answers we were looking for. And so it was a two-part survey because as they went through the screening survey, they basically couldn't then, once they submitted that screening survey, they couldn't go back and change their answers. Versus if it was all in one survey, you could go back, you can always go back and forth within REDCap. And so once they submitted basically the screening survey, which assessed for the inclusion exclusion criteria, there's basically a hidden field variable within REDCap that determined their eligibility. So it's a computed variable, basically, adds up. If the score is 8, then therefore they're eligible. And so if they were eligible, then they got basically a follow-up email, which had the, thank you, blah, 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 and had the URL, you're eligible for enrollment, and here's the link, here's the URL link to go back into REDCap for the enrollment survey. And those who basically the computed variable didn't add up to be eligible, they got a thank you very much email, but didn't have the URL for further enrollment. And so this sort of two-part survey, within this there was sort of logic and we sort of mapped this out as based on the questions, were people going to be eligible or not eligible? And so every sort of box, needed like some answer of were they going to be eligible or not. And so there were sort of slightly different criteria. And so basically we sort of used this branch logic to do this. So I'll kind of skip through this. But one example, like for instance, for healthcare workers, if they were were negative at baseline, were they symptomatic or not symptomatic? And if they were symptomatic, how long were they symptomatic for? And so were they eligible or not eligible for the trial? And so for enrollment, we use an e-consent process via REDCap, and so that's sort of built-in function. It adheres to sort of FDA guidance. There's a guidance document that was published in December of 2016 on e-consent. And importantly, I think we just didn't have like, here's the consent form and click here and sign, because I think anyone that's done some of these, agreements for Apple or iTunes or whatever, like you have some 20-page form and you sort of scroll to the bottom and click on it. That doesn't necessarily mean you understand the consents at all. And so we did have a series of questions. I think there's 5 questions basically to assess their people's comprehension, sort of big picture of like, what do they need to do for the trial? What were we asking them about? There's a multiple choice. It wasn't exactly a really difficult quiz, but we wanted to make sure that people had some comprehension of what they were actually consenting for. And they actually read the consent form. And so they failed that quiz. They had to go back and basically reread the consent form to make sure that they were their comprehension was adequate. And so within this, and so if they passed that, we then collected their email, their address, their phone number. Obviously, that was for shipping reasons, but also for follow-up. And then a next of kin contact. And so we stated if it was hospitalized, but also if they were lost to follow-up, if they died, who could we basically contact? We weren't quite that morbid in our questioning, but we basically asked if they were hospitalized, who could we potentially contact? And so all these pieces of information were also sort of ways to sort of vet people, to make sure that they're actually providing truthful information. So if they're have a 555 area code number for their phone number, like, hey, that's a fake phone number. If they list themselves as their next of kin, like that's not great either. And so we did sort of screen people out through that process. Some of the things that we should have collected, which we didn't, was if the phone number was a mobile or a landline, and their preferred follow-up method. And so you can send out text messages from REDCap that's integrated via a Twilio sort of function. And so versus email. And so I think we all get like tons of emails every day, and just some people just delete them or they just kind of blend into the background, versus some people like text messages because for a short survey you can just kind of click on the link, boom boom boom, answer a couple of questions, and that was easier. And then the last thing we should have probably collected, which we didn't at enrollment, was their social media contact information. So nowadays people are on Twitter or they're on Facebook, and so this is probably a much easier way to assess their vital status than actually having to call people up, because people don't answer their phone. And so if people are posting on Facebook or Twitter like, hey, they're still alive. And so for those who are lost to follow up, when we're doing an intent to treat analysis, like, hey, are they alive or not? This was probably could have been an easier way to assess vital status instead of calling people up, the 10% who were lost to follow up, to try to figure out their vital status. And so, the other thing is having a 2-part enrollment, the screening, where they had to enter their email address, verified that their email actually was a working email and they were able to receive it. We did warn people, sometimes people had spam filters or blockers and stuff like that. So to make sure that the email address would work appropriately. And so that was also part of the reason for the 2-part sort of screening, then enrollment. So our screening surveys, as you might remember back in April, hydroxychloroquine was in the news a lot, and the president was talking about it, and it was sort of a big just mess, is what it turned out to be. And so these are just the number of our screening surveys. And so the first day we screened 49 people. One day it was up to 1,400 people, when basically on WeChat, sort of a Chinese language social media platform, people were having conversations about this, and sort of word spread. And so things sort of varied. I think letter C on my graph is I went on Dr. Oz and talked about this, which my wife hated. She's another physician, and she was really disappointed that I went on there. But he has a big outreach, and we got a bunch of people screened, and some people enrolled too. So it was great. And then letter sort of E and F were basically when negative media came out on. There was a chloroquine trial from Brazil, as well as a VA study. And then F was when the FDA put out an announcement that hydroxychloroquine could be dangerous, and there was a caution released on April 24th. And after that, basically killed our enrollment. And even if we wanted to enroll more people, our trial was pretty much done at that point. So within, we excluded our trials in total enrolled about 3,000 people. And so for our PrEP and early treatment trial, which enrolled about 1,200 people, we excluded over 7,000 people. And so a lot of that, if you had to call or you're meeting people in person, that would take a lot of coordinator time and a lot of effort. And so the pilot, sort of the logic by the automated sort of internal survey was able to sort of exclude people. And so the major exclusions were either people didn't have a PCR test or didn't have contact to someone with PCR test, or they were symptomatic for more than 4 days. And so for our early treatment, they were out of the window for that. And so that accounted probably about 95% of the people who were sort of external were excluded from the trial. And so one recommendation is we did pilot the survey sort of questions and logic before we launched the trial with both medical people and non-medical people. So particularly the non-medical people of like, does this language make sense? Is this sort of appropriate lay language that non-healthcare folks would understand? And so people had some good suggestions on modifying our wording. We also had sort of incomplete enrollment forms. And so we did have someone manually looking over the forms to make sure that they were complete. And so even within REDCap, where you force a field that has to be completed, doesn't mean it has to be completed correctly. And so people could just, if it's a text field, they could just put in garbage. They could just put in, just nonsense stuff or phone numbers that, yeah, you have, the right number of digits, but it doesn't make sense as a phone number. And so, so we did have people, review for validity. And then we also looked for duplicate screening forms. And so if people were sort of fishing like they were excluded and they went back and they changed their answers to like try to be included in the trial, like we were looking for duplicate forms. And so those were flagged. And unless people, emailed or provided some explanation, we did not enroll those people. And then as well, like as this trial went on, we continued to sort of evolve and update our logic to make it slightly better. And as more information was known about COVID, or as some of our inclusion/exclusion criteria were tweaked based on FDA feedback, there were continual reviews of this. And so as we would update stuff, sometimes people would slip through the cracks. And so noting sort of as you update things to make sure your logic is still working. And then as it was an internet-based trial, like anyone could log on. And so we had one person that tried to enroll from Mexico. And I was like, oh my gosh, we didn't enroll that person, did we? And so we didn't, fortunately, but you realize like, oh, you might actually get more people from destinations that you were not intending. And then we had some people that were just like outright sort of fraudulent and trying to sort of just kind of figure out our survey and our inclusion, our logic, which is like, if you just would have asked us, we would have given it to you. So overall, sort of this sort of the REDCap sort of survey and logic worked pretty well to sort of screen out the vast majority of people who were not eligible, and really reduce the amount of work that was done. So for logistics, how do we actually do this? And so from REDCap, you basically can export your data set. So we had sort of a report within REDCap of the variables we wanted to export. From that, we used a mail merge function in Word to generate a prescription, which was a PDF that got sent by secure email to the research pharmacy. They were randomized at the research pharmacy. And then they basically made up the prescriptions that were blinded. And then at the same time, we were then using the same sort of Excel sheet to import into a FedEx Ship Manager software, which then generated the FedEx labels to ship out to people. And so we basically shipped out Monday through Saturday for next business day delivery. And so then we basically picked up the medicines from pharmacy, and we had a bunch of our investigators stuffing envelopes to then drive it out to the FedEx drop off point for the last sort of shipment of the day. So so that was sort of the only manual aspect, was stuffing envelopes. And so I stuffed more than my fair share of hundreds of envelopes, but that was fine. But the process was fairly automated, so the actual amount of work was pretty simple. So when we looked at the time from enrollment to drug delivery, one of the things that was impressive was that about 2/3 of people actually enrolled outside of weekday sort of business hours. And so a lot of people were enrolling at night or on the weekends. And so if you have sort of a working, we enrolled a lot of healthcare workers who were working. And so, and just when people had time, that a lot of times when people are surfing the internet, I guess, they find out about like, oh, there's a trial I could participate in. This wasn't necessarily during the daytime hours. And so I think that really helped our enrollment because if we're just trying to reach people, if you're trying to call up people like, hey, you have a positive COVID test, and trying to reach them during the daytime work hours, either people won't answer their phone, or they might be annoyed, or they may just not want to do it because they're busy. And so having sort of an internet-based sort of portal for entry, I think helped our enrollment. And the peaks here, basically the first peak are people who enrolled in the daytime, sort of before 4 p.m., which was our sort of cutoff for pharmacy. The second peak, so they would have basically gotten it, the next morning at 10 a.m. The next peak is people that enrolled in the evening that then, would have been randomized the next day and then shipped and sort of have a 36-hour delay until they receive the medicine. The sort of the little small peak there is sort of Saturday morning or on Sunday, or it could be up to 2 days, where if you enrolled on Saturday morning, we shipped it out Saturday, but you wouldn't have gotten it until Monday morning. And then the last peak, was basically up to a 2.5 day delay. And so the worst case scenario was if you enrolled on a Saturday PM, then you didn't get randomized by the pharmacy until Monday, and then it got shipped out and arrived on Tuesday. And so that group, we used a little bit of discretion as far as would they meet the time, would they meet the window for trying to get them drug within 4 days or less. And so that group we probably could have excluded in retrospect, just turned off our screening survey on Saturday. Would be one way to shorten that. But then they could have just enrolled on Sunday or on Monday and still had the same effect as far as the overall time. But that was one delay, which was known, but and we could have done something about that. But we basically looked at it as far as what their time was from enrollment, and so after they enrolled, they basically got follow-up emails on the next sort of business day. So day 1, day 3 was if they were symptomatic, but we eventually just changed that to everyone. Day 5, day 10, and day 14. And then the PrEP trial used weekly messages where they basically got a weekly message to assess their follow-up over the 3 months of follow-up. So overall, I'd say about 75% completed the follow-up surveys really well. And so we didn't have to prompt them. They didn't need any reminders. For those that missed a survey, then REDCap would basically send out another automated sort of email the next day as a reminder. And about 15% of people needed sort of additional prompts, whether it was the follow-up emails or text messages or phone calls. And then about 10% were really lost to follow-up and just didn't respond to anything. And so some of the things that we did do within the study, which I think was kind of interesting, was do remote blood collection. And so we did do a PK study. We did a serology study by Melanie Nickel that led this on our team. And so we used these sort of blood collection kits where they come as these kits. Within the kit, there's the lancets, there's Band-Aids, there's the collection tubes, which basically you stick your finger and they sort of suck up whole blood and sort of various quantities, whether it's 10 microliters up to 30 microliters. So a small amount of blood, but enough to do a PK study or do a serology study on. And these sort of kits are kind of nice. They're sort of all packaged and together. They cost about $25. Obviously, if you're shipping stuff, back with FedEx, they need to be stable for at least 24 hours. They need to be temperature stable. But most things, can be, especially if you're looking at, chemistries or something like that for safety labs. And so you could do remote monitoring of safety safety events even within sort of this internet-based trial protocol. So this is kind of cool. And then other people, we didn't do this, but other people at University of Washington did do self-collected nasal swabs. And so they basically had the same thing where it was, boxed up and came with some nice instructions as far as how to collect anterior nasal swabs and then send those back for virologic testing. And so this is pretty cool. We didn't have the money for this, and we asked for money from NIH, and we didn't actually get any. But we wanted to do this, but at the time back in March and April, there was a shortage of swabs and there was a shortage of testing. There was lots of problems. And so in hindsight, this would have been great, but we didn't end up and do this, but others did. So endpoints, for clinical trials, a lot of times thinking about your endpoints up front is obviously very critical. And so for ours, we use basically patient-reported outcomes. And so you think about clinician-reported or patient-reported outcomes, or you might have a lab outcome. So in this case, our things were based on patient-reported outcomes, and basically for our post-exposure prophylaxis trial, it was like, are you experiencing COVID symptoms? And so they said yes, then it was like, okay, what was the checklist of going through sort of the fever, cough, shortness of breath, and going through sort of the 12 different symptoms that CDC reports, and also providing a free text so they could elaborate if they wanted to talk about other things. We also use a visual analog scale to report the overall severity of symptoms, and I'll show that next. And then we also asked about side effects. We same thing of having a checklist of what are the common sort of top 10 side effects of hydroxychloroquine, and then having a free text field as well. So this worked well because the medicine had a pretty well-known safety profile. If it was a new medicine where you didn't know what the safety profile was, then this would be a little bit more problematic. You'd have to do much more extensive aspect of that. And then we asked if people were hospitalized. That was an endpoint for our early treatment trial. And then we asked about sort of a targeted list of medicines on day 5 and 14. And we didn't list about every medicine they're taking, but we had, were they taking zinc or vitamin C or azithromycin? So we had a targeted list of medicines that we cared about. And then at the end of the study, we also assessed for the adequacy of blinding, asking what do they think, which treatment arm were they assigned to? And it was pretty, revealing where the people actually, the blinding worked fairly well. We used folic acid as our placebo. And of the people who had side effects, they thought they got the active drug, whether they had side effects through the placebo or whether it was the hydroxychloroquine. And the people that didn't have side effects, actually, it was a pretty good blind. And so that worked well. And then, so as part of our patient-reported outcomes, we basically ran through the list of symptoms that were part of the U.S. sort of case definition. This is sort of the CDC working definition. And importantly, because we were doing early treatment trials, even, people could have false negatives PCR tests, because if on the first day of symptoms, the reported rate is up to 38% of people can have a false negative PCR. And if you continue to test, then it'll be positive. And so, so we kind of, looked at both of these as far as, lab testing, as well as did they have a compatible illness? Because pretty much everyone had an epidemiologic linkage, that they either, for a post-exposure prophylaxis trial, we required people to have a exposure to someone who's PCR confirmed, or in our early treatment trial, similarly, like the majority of people needed epidemiologic linkage or their own positive test. This definition worked out okay. And then the question is, well, do you need to see a patient, or can they tell you, like, oh, I have cough, I have shortness of breath, I have different, a lot of these are really subjective sort of things that, if someone has a headache, there's nothing on physical examination I'm going to look and see to verify they have a headache. And so a lot of these, I think, are, legitimate, patient-reported outcomes. And as we reported in the appendix, like the sort of what was the symptom complex of these cases, and it was sort of all over the place as far as a lot of sort of the probable cases. We also asked about other symptoms that weren't sort of COVID necessarily related as well, just to keep people a little bit honest, I guess, and ask about other things. And then we had possible cases and non-cases, and all the case adjudication was done blinded. The visual analog scale that's in REDCap, it's sort of a built-in function. We basically asked sort of what's the overall severity. One thing we learned that like people might have a 5 and they just kind of look at it and say, okay, it's a 5, but you actually have to physically click on the button to make it a 5. So you actually have to click on it and move it or leave it where it's at. Otherwise, it's recorded as a missing variable, missing value within REDCap. And so this is totally a subjective measure, which we acknowledged. But what we really cared about was the intraperson change over time. And so how quickly did people get better with the intervention? And so that, I think, is really quite valuable, because ultimately, in medicine, the goal is to make people better. And so making people better faster is the goal. And so, different people may have different sort of baseline severities, but ideally, people are going to get better over time. And so there's tons of publications. Oftentimes, this is a pain scale. People use it for cough severity, other things as well. But this is a simple function that was built into REDCap, and we kind of liked it. So what are some of the expectations for future trials? So from our early treatment trial, just to show a couple things, as mentioned, we targeted either PCR positive persons or symptomatic persons with epidemiologic linkage to a PCR positive person, because this is once again back in March and April, where outpatient PCR testing was really quite limited in lots of places in the US. Even like healthcare workers who were exposed and symptomatic, they couldn't get testing, which was like, what? How can you not get testing? But that was the case. And then a lot of times the PCR results were delayed. And so people would have like, I have a pending test, but it's not back yet. And so we basically had to make a decision. And so as we designed the trial, we basically focused on, people that had a positive result or this epidemiologic linkage to occupational exposure or household exposure. So if, one person has a diagnosed COVID in the household and the other person has fever and cough and like, COVID symptoms, like, what's the probability they have COVID? It's probably really quite high. And so overall within our early treatment trial, about 80% of people were either PCR positive or contact PCR. The other ones basically were enrolled on a pending PCR and their PCR came back negative, or some of them got lost and they never got the trial results, but had sort of made 2 of 3 major symptoms of fever, cough, or shortness of breath. And these were sort of a priori subgroups that we had identified that, hey, we realized that, maybe some of these people didn't have disease. And so we were going to look at the response across all of these three groups. And so importantly, the people with lab confirmed disease, they were slower to enroll. So it takes longer to get a lab test back. And so the mean duration of symptoms for people with lab confirmed disease was 2.2 days. For those that enrolled via sort of a contact being PCR positive was 1.3 days. And those pending cases with a pending PCR that were enrolled based on their symptoms of two of three symptoms was 1 day. And so overall, we were able to enroll people fairly quickly. So 56% of people enrolled within 1 day of symptom onset. And so we were trying to do early treatment. And so I think as people think about early treatment trials, most of them are requiring PCR confirmation. But then the average time of when they're enrolling is 4 or 5 or 6 days into their illness. And so a lot of these treatments are going to work much, much better if they're given early. And so I think that's one sort of negative, I think, of some of the trials that are being run is that, if you're treating people at day 6 or 7, by that point, their illness may already sort of, whatever is going to happen is going to happen. And so you may have less room for intervention there. And so, but once again, we define these as a priori subgroups. And so if I was going to run another trial again, I would do the same thing of enrolling these people, but I would then send out swabs to confirm the diagnosis for people that were enrolled via an epidemiologic linkage or had a pending PCR, other things. So overall, not to focus all on the adherence, but 2 points. One was about 15% of people basically immediately were either lost to follow-up or reconsider their participation in the trial. And so I think that's something, as you do an internet remote trial, you realize, like, you ever bought something on Amazon, like you buy it and then like 2 days later it comes, it's like, shoot, do I really want this? And so I think there's a buyer's remorse sometimes a little bit. And so some people like reconsider, or they take sort of, there's other influences, or maybe they get a different treatment or something like that. And so some people may unenroll in a trial. And some of these people just were lost. They didn't really withdraw consent per se. And the other thing is FedEx, about 1 to 2% of packages were lost. And so they were misdelivered, they got stolen, they just, people moved, they were quarantining. And so there was some people that just didn't get the medicine. And I think that's sort of just part of the deal of doing sort of business. And so we did an intent to treat analysis. In retrospect, we should have done a modified intent to treat analysis and exclude the people who never started to study medicine. We didn't realize this was going to be quite so high, but part of the not taking any study medicine was also the people who were lost to follow-up. So that 10% was sort of captured in there. So, and then obviously this study was not operating in a vacuum. And so there was substantial media coverage and things changing over time and people having really strong opinions, most of which were basically not warranted. The symptom duration sort of abated over time. And so as you think about other COVID trials, sort of knowing the natural history is really important. This was, we didn't know at the time, when we started in March. And the hospitalization rate was much lower than anticipated. It was only 3% in our placebo group. It was 2% in our intervention group, not statistically different. And then the change in symptoms from baseline. So we used this visual analog scale and looked at sort of the change over time, and it sort of came down both similarly in both arms. And so this sort of change over time metric was statistically a much more powerful metric than if you look at a categorical outcome of yes, no, hospitalization. And so this is what we ended up and changed our primary endpoint to. The change from baseline was normally distributed. The absolute scores were not, but you could log transform those, and we did that as a sensitive analysis. So some of the good things or bad things about our trial, it was one of the first to really initiate and complete enrollment. We enrolled nationally across, everywhere in the U.S. virtually. Some of the limitations, because it was remote, within the resources we had, we couldn't do PCR confirmation, but you certainly could do that of sending out nasal swabs now. That testing was availability. And we sort of designed it knowing that testing was not very available. And so that was sort of one of the things. So what are the roles of virtual trials? And this is going to be where I stop here. If you can use patient-reported outcomes, so that's certainly ideal. Otherwise, like the clinician-reported outcomes, so the UK recovery trial that's basically across the NHS is using clinician-reported outcomes. You need a drug or something with a good safety profile ideally you don't need laboratory monitoring. You certainly could do remote laboratory monitoring, but it'd be much more complex. Or you could do a limited subgroup with safety monitoring, and then a larger group without safety monitoring. So it's in general, I would think this would be really best for sort of rare and neglected diseases where you've got a really motivated participants that want to come and like help participate in a study. Ideally, something like a chronic disease, which is less acute, where you got less of a time pressure, would be great. I think would certainly be a role for for rare and neglected diseases. And then strategy trials of therapy A versus therapy B that are both FDA approved therapies. Is that the best? Which one's the best? And so for these strategy trials, they're never going to get funded by NIH or a pharmaceutical company usually for our already approved drugs, but we want to know like what's the best way to do something. And so this is really an endless amount of things. And so can you do a study really for a low cost where the only thing that really for us was shipping, basically FedEx shipping, and was personnel time. And so that was really the major two costs for our study. And so I think that virtual trials may certainly have a role for strategy trials and for the rare and neglected diseases. So we had some great funders that came to us and basically gave us money, which that's never happened to me in my life, where people come find me. And so this was funded by a variety of sources. It started out with just myself and our university as a land-grant university. And we had local organizations and then other foundations around the country. We also applied for NIH for emergency funding, and we were unsuccessful in 4 different applications for the 3 trials. So, and then a lot of the personnel were covered by other sources: training grants, or career development awards, Doris Duke Awards, Fogarty's as well. So that was great. And ultimately, we had really great team science. And so that was, we had, med students, ID fellows, a lot of these people had worked on our cryptococcal trials in the past. And so they're pretty experienced. And we all had a lot of free time because basically our research was shut down. And so that's what we pivoted towards. And we ran these trials for about 3 months, and then our research has started back up. And so we've pivoted back to our normal research. And I've gone on to provide advice to people, but that's mostly I'm back to my fungal world of things. So thank you very much for the invitation to speak. Hopefully you learned a little bit about how we ran our trials. And as you think about other sort of virtual trials in the future, post-COVID, I think that this really could play a role of ways to do sort of low cost, impactful trials. And happy to take questions.
So David, this is Rebecca, and one of certainly the lessons you learned. This was an enormous amount of work in an incredible time frame, and when you traditionally think about this probably would have been, what do you think, 6 times longer?
Yeah, I mean, if we only enrolled in Minnesota, like one of my friends is doing an ACE inhibitor study, and they just wrapped up. So they basically enrolled for like 9 months to get, similar or actually probably smaller sample size. Just because if you're only enrolling locally and it's only, your lab and you're trying to call people in a very traditional way, you're getting people, like if you're doing outreach to try to get the people into your trial, like they may or may not be interested, versus in an internet-based trial, like they're coming to us, they're already motivated to participate, and they've at least thought about it a little bit. And so I think that was helpful. And so then it's really about getting the word out in a realistic way to refer patients.
So I think that one of the great opportunities here is to really kind of take all of these methods that you did, lay them against some of the other approaches that people have used, particularly in these kind of rapid onset trials, and to look to see where there are consistencies that are things that we could adopt or change systems for. Because I think that we certainly put sometimes in that desire for safety and quality, sometimes put barriers that in fact your data would suggest aren't maybe as critical of an issue when you're very carefully selective in the types of studies that you do, and then kind of take this kind of new sets of methods and really apply it to a rare disease trial, which would be absolutely ideal. I think about it from my interest, like osteogenesis imperfecta. Community very quickly, even in a cold call survey that we did as part of a PCORNet study, had, like 65% return on surveys. Us literally, sending them, a very, very much too long survey in order to understand things. But I do think that these are the kinds of things that we need to think about, and that in fact, this pandemic really has allowed us to move forward in the future.
I think particularly for rare diseases, if there's an organization, because the question is, how do you get the word out? And that's really the challenge. And that was probably half my time was like doing media stuff and sort of trying to spread the word. And so if you already have an organization, and a lot of these rare neglect diseases do have organizations that are sort of a focal point. And so if you have a way to get to that patient population to let them know about stuff, then I think the possibility of doing trials becomes much more likely. Any other questions? Well, Dr. Jackson, thank you very much for the invitation today, and hopefully
thank you.
People learn something.
David.
Okay. All right.
Hi, everybody. I'm Tim Worth. I'm the Director of Biomedical Informatics for the CCTS and the CRIO of the College of Medicine and the Wexner Medical Center. It's my pleasure to introduce our next set of presenters. Doctors McIlerney, Frey, and Panchal are part of the investigatory team to establish the Center to Stop COVID. Anne's office is actually across the hall from mine in IBMR and was the singular person to recruit me to OSU. We've been longtime friends, and it's my pleasure to introduce her. Anne Sheck McIlerney is a distinguished professor of family and community medicine and the executive director of Catalyst, the Center for the Advancement of Team Science, Analytics, and Systems Thinking in Health Services and Implementation Science Research in the College of Medicine at The Ohio State University. Anne has over 30 years of health services research experience, during which she's been actively involved in both performing research and disseminating research results to academic and practitioner audiences. In addition to co-leading the Center to Stop COVID at OSU, Dr. McIlerney's ongoing research focuses on information technology innovations in healthcare, population health management, quality improvement, and organizational development. And she's an expert in both qualitative and mixed methods analyses. Dr. McIlerney received her undergraduate and graduate degrees from Stanford University and Harvard University's T.H. Chan School of Public Health. Dr. Jennifer Frey is a clinical research manager for the Department of Emergency Medicine at The Ohio State University. In her current role, Dr. Frey co-manages a clinical research staff which engages in research within the two OSU emergency departments. Dr. Frey's work has included program management for the Center to Stop COVID, coordination for a clinical trial comparing known treatments for severe infections called the CLOVER study, observational study to understand the pharmacogenomics of opioid use, and implementation of a clinical program to initiate medication-assisted withdrawal in the emergency department for patients experiencing withdrawal from opioids. Dr. Frey holds a bachelor's degree in molecular genetics from The Ohio State University and a PhD in experimental therapeutics from the University of Texas Health Sciences Center and MD Anderson Cancer Center. And finally, Dr. Ashish Panchal is a professor in emergency medicine and is currently heavily involved in pre-hospital care, being the medical director for Delaware County's EMS and the Delaware County Emergency Communications and Delaware County Emergency Communications, as well as the associate medical director for Worthington Fire Department. At The Ohio State University, he is the program director for the ACGME accredited EMS fellowship. And on a national level, Dr. Panchal is the research and fellowship director of the National Registry of Emergency Medical Technicians and is part of the expert panels for the National EMS Scope of Practice Model Revision Project and the evidence-based guidelines for pre-hospital naloxone administration and the EBG for pre-hospital pain management. He also has a strong focus on EMS provider wellness and has conducted many studies describing the associated risk with pre-hospital care provision. Their presentation, pivoting priorities and responding rapidly to create the Center to Stop COVID, will discuss their experience. Dr. McIlharney.
Thank you, Tim. I'm not sure how this is supposed to work, but I'm assuming I can share my screen and get this going. Okay, look good. Thumbs up from somebody.
So I'm
All right,
seeing
let's
your
see.
presenter view rather than just the slides.
Okay, in that case, we don't really need that.
And click swap displays. Top left-hand corner.
Top left-hand corner. Ah, there we go. Thank you. Tim is also my IT support in every aspect of my world. All right, so Tim told us the title. This is our presentation. We are delighted to be invited and wanted to give you just a little bit of an overview of what we plan to present. First of all, we're going to talk a little bit of our prior experience and the research that we needed to pivot. We'll provide a little bit of background about the considerations and the context of COVID-19. And then we'll go ahead and talk about our Center to Stop COVID in terms of the embedded research projects, our project goals and deliverables before opening it up for discussion. So to start, the Center to Stop COVID stands for the Center for Serological Testing to Improve Outcomes from Pandemic COVID-19, or Stop COVID. We really like acronyms, and Ash was able to put this together to say Stop COVID, which we really liked. And as you can see from the now ubiquitous Zoom pictures, this is a picture of our team members celebrating receiving our multidisciplinary grant from NIH and NCI. And there's animation that kind of shows how the different agencies have put together. This is actually part of a broader serological sciences center of excellence. And Ash is going to talk a little bit about that next.
Yeah, so overall, the SeroNet includes representatives from the National Cancer Institute and the National Institute of Allergy and Infectious Disease. Additionally, it includes all of the U54 sites. That's all the sites here on your left-hand side, from anything from North Carolina and so on and so forth. And you can see a couple of more fun little acronyms here and there. But it also included all the U01 applications, which are on the right-hand side. Now, added to this, you also have the Serological Sciences Network Coordinating Center and whoever got those awards. So overall, now you have a large group, SeroNet, which includes essentially people from all over the country who are looking at the same question. And it's the concept here is they wanted to build a strong, transparent sharing of data, assays, samples, references, so that we could do all this research together and be truly impactful. Next slide, please, Ann. So one of the key things that I really wanted to highlight which was our hurdle and I guess our success of this application is really apparent in this timeline. I'm going to take you over this timeline, but when you think about grantsmanship and getting grants in, if you look at this timeline about the point of when we started hearing about stuff and when it got funded, condensed is really an understatement. So when all this started, it started with groups forming in late February who are looking at convalescent plasma theory, antibody testing, COVID research, and you can see the key individuals who are helping move this forward. Now that was in late February, and then the RFA was first released in the beginning of June. And so that was the emergency RFA. And Dr. Ollis has really started looking around for a project or a group that would make OSU stand apart as we put in a U54 application. Now, as June continued, this is when we started really getting the teams together, and Drs. Lozansky, Bachman, Lowe, and Castillo were able to sit down with Gene and really talk about a possible project with first responder surveillance, since they're such a high-risk group. And this really struck a chord with the team, and we started assembling larger groups and on Zoom calls. And that's when Ann, myself, and Linda Safe were all added to the team. we actually started writing the application in July, and we're done writing this enormous 600 odd page application by like almost the last week of July. And so the condensed timeline was amazing, but the very fact that we did all this by Zoom and we never hung out or met or really had any type of cohesive team forming, it was all these discussions together and pulling the team together, which really moved it forward. We got our score in the end of August, and we were funded by mid-September. So this turnaround and how this all came together for us, and I look back myself and think about this, was one of the most phenomenal pieces of work that we've done in the sense of really coalescing a team. And a lot of credit goes to Jean and the other MPIs and how to put this all together.
So, hi everybody, I'm Jennifer. I'm going to talk about the prior experience of our MPIs and our directors. And to tack on to what Dr. Panchal said, is we have this incredible group that came together and has yet to meet in person. And it started with Dr. Oltz leading as an internationally recognized immunologist. He's been at OSU for several years, and he's come with lots of NIH program experience and chairs the Department of Microbial Infection and Immunity. And he, along with Dr. McIlerney, who is the director of Catalyst, which is a health services research enterprise here at the institution, were able to really continue on building this group. And Dr. McIlerney comes on her own great experience of funded research, looking at health services and leading large P30 type grants and many, many others. Dr. Panchal also joined the team as a clinician scientist who brought in that great expertise of emergency medicine services, pre-hospital care, and is also a very well-established clinical and outcomes researcher himself, looking at pre-hospital care and resuscitation and airway management in the pre-hospital environment. He also has a lot of leadership experience in as a research fellow director. And then if we go to the next slide, Dr. Safe is just such a pleasure to be working with. She's an internationally known virologist. She is a member of the National Academy of Sciences, and she has very close to 400 publications and over 75 book chapters that she's published. And her expertise in coronavirus and other viruses is just leading all of the fundamental research in terms of the disease and the serology. And then Alice Gahn and myself have supported the program as center administrator and the clinical research manager. Alice has worked with Dr. McIlerney for many years, working on managing multiple research projects. And she's now the assistant director of Catalyst. And then I have 10 years of emergency medicine experience that has been tacked on to my early research in the vet biosciences area and retrovirus research. So this group together has built this very large, and this is only the tip of our team iceberg of expertise that have built into the study.
Thanks, Jen. So just to continue, some of the things that we really had to consider, in addition to some photos of our other MPIs that are perhaps on the call but not visible, we had to figure out how to leverage Linda's prior work on coronaviruses. She is the world's leading expert on coronaviruses writ large, and had been working and thinking in this area, and really is the serologic expert in terms of this group. So we had to figure out kind of how to showcase that as we developed this large application. For those of you that haven't put together a U54, which used to be me, the idea behind the U54 is it is embedded research projects that are individually the size of R01s, supported by cores. And so we really wanted to embed one of the projects to focus on Linda's contributions. Another thing that we had to consider was how to work with the first responder populations. So these relationships, unbeknownst to me, had been really well established, both with the Columbus Department of Fire and the Department of Police. And these relationships then had to kind of coalesce in trying to figure out how we could work with this first responder population. And that became part of the project, the first project. And then the third project was the big question around how to embed a health services research angle as a project, given the expertise of me and my colleagues at Catalyst. This was probably the most out of the box thinking for our project, just because the emphasis on the RFA was really around the science. So it was really around the bench science, the lab science, some clinical science as well, but thinking about how we could look at health services research angle, implementation science angle. And that's where we started to think about some things around Project 3, looking at risk communication, looking at communication across the entire project, and looking at really how we can learn from the other projects and then the broader community, scientific community as well. We also were looking at how to leverage the clinical trials expertise. Jen and Ash have been doing this for a lot longer than I have in terms of actual patients and blood and saliva and that kind of thing. And we were able to kind of pull the words and processes together to make a compelling case that this is something that we could do and hit the ground running. And going back to that timeline, we were funded in mid-September, and the idea was you have to start the next day. So there wasn't some big old gap. We really had to get going. And then the last thing, that one of the last minute things was, how do we justify 4 MPIs? And we just collectively decided they might like it, they might not like it. So we went ahead and submitted. The other thing that has been emphasized, I think, from all of these projects is the notion of COVID-19 provides this really funky context. As Ash mentioned, we have not all met each other in person. Linda is actually up in Worcester, and I'm pretty hard pressed to even put Worcester on a map, although I'm really excited to go visit Linda when this is all over. In addition to that, we have COIs that are distributed across 2 OSU campuses, 5 different colleges. All of the COIs have their own different research labs, their own different processes. We have basic sciences, we have health services, we have clinical sciences. All of these things are very, very different and distributed. So just from a logistics standpoint, trying to put together the letters of support across the departments by the different divisions, colleges, think about putting together the PA 005 with this enormous allocation challenge in terms of trying to figure out how the colleges got and the departments and the divisions things. And then on top of that, we had power outages and submission glitches, and we had long sleepless nights where when we submitted the application, it didn't actually go through. And so there was some scrambling by our grants management people to go to campus to try again that way. We left that night thinking, we made the deadline on our end, but we didn't get that receipt until the next day. So I think Jean in particular probably didn't sleep at all. I happen to be a good sleeper, so I still slept. I was just anxious. And then we found out that with our cover letter and everything, they were able to accept our application. So this all came together in our Center to Stop COVID. And Ash, am I supposed to give to you? I've kind of gotten lost now in my order.
No, I think
Still you, Ann.
Okay, still me. Okay, great. So it is a SARS-CoV-2 Serological Sciences Center of Excellence focused on investigating the interface between exposure risk, transmission, immune responses, COVID-19 disease severity and protection with a goal of improving clinical outcomes and population health. That's a mouthful. Anyway, this lists out kind of how the organization is here, so that we have Core A, which is the administrative core. Core B is the testing and biorepository core. And then Core C is the data management and analysis core. Also embedded within this is that we have leadership and stakeholder groups. So we have our executive committee, which is the MPIs, and the directors. We then have our center steering committee, which is meeting still weekly, which is the core and the project leads. We have our internal advisory board, which is consistent of institutional leadership. Then we have an external advisory board with community partners. And then we have the Serological Sciences Centers and the Seronet. And the Seronet actually got up and running remarkably quickly. And they have actually been having periodic meetings. There's another one this week. So in addition to kind of what we're doing, we're also keeping a pulse on what the other places are doing so that we can kind of leverage what they're doing and kind of think about it that way. So the opportunity with the Serological Sciences Centers and Seronet is also to participate in operational boards, which have started to meet. So for instance, I'm on an operations committee that's looking at communication. And of course, the vaccine information is the biggest topic of communication right now. So trying to figure out where that goes. And then the 3 embedded R01 research projects at the bottom there, project 1, 2, and 3 led by different people. And I'm going to skip over that because we're going to talk about those in greater detail in the future. So where we are is the work has begun. We had to have IRB approval to receive funding. That was a little bit of a dance to try to figure out how to actually piggyback on existing applications, but make the proper amendments and so forth. We have amended and modified different protocols as the pandemic has evolved. For instance, for Project 3, we've had to add questions about vaccine hesitancy, which we did not anticipate. In the beginning of when we first submitted for our IRB approval, we were asking, you know, hey, what do you think if a vaccine becomes available? And now that question line has become a little bit more complicated. We have established a project website where we're promoting what we're doing. As I said, we're considering how to incorporate the vaccine information, and we're starting our first responder enrollment. Is it this week or next week? I said next week, but I drafted it.
It'll be next week.
Okay. I got lost on my own timeline. So with that, I'm going to turn this back over. This is our big overview picture, but then I'm going to have Jen talk about Project 1. So this again is just a different picture of what we're doing with Project 1, Project 2, Project 3, the different cores, and then some cool pictures. So Jen, why don't you tell us about Project 1?
So as I mentioned, like the MPIs and the coordinator leadership that I mentioned are really just the tip of the iceberg, and there are so many other people involved. We have co-investigators for this project in particular. We have Dr. Dan Bachman, who's an EMS, disaster medicine, emergency medicine expert. He's also led a lot of what our community has seen in terms of COVID swabbing at our swab stations out in the Buckeye lots and at the Slush Center. He's instrumental in all the logistics going on there. And then Dr. Gerard Wazansky, he is also a clinician, pathologist, and working on the testing in the clinical setting. In addition to Dr. Oltz, who's chair and the expert in immunology. So the goal of Project 1 is to look at the duration, protectiveness, and antibody responses of our Stop COVID participants. And these participants are going to be our Columbus Fire and Columbus Police first responders, as well as their household contacts. And we're going to look at, we know that they have been heavily impacted by COVID in the community. We've had entire fire stations that have had to put everybody on quarantine based on exposures and things like that over the last 9 months to a year. So this is part of the justification that makes this the most excellent group to study. In New York, they were having back in April, 5 to 10% of their first responders were coming down with COVID. So the project is really going to look at that asymptomatic antibody response and viral presence in our study population. And we're also going to be collecting samples that are going to go to our Core B biorepository so that they can be used for other testing of other studies. We're also going to be looking at the serological responses in our subject population after they get vaccine, or if they are infected, or if they've been previously infected. Go to the next slide, please. Exciting. I like this slide. This really actually helps. We are going to be testing our subjects every 6 months to look at that viral and serological presence. So we do have this as a 5-year longitudinal study. The one of the other fun caveats of this study, and this is what's made it such a interesting study to set up from a clinical trials perspective, is if subjects become symptomatic or COVID positive, they're going to enter a symptomatic pathway. And this will allow us to do a deeper dive into that serology and viral presence as they're experiencing symptoms. And so we will follow them for up to 25 days after they've experienced symptoms and perhaps become PCR positive through testing. And one of the biggest caveats of setting up the study and the need to continue to pivot is we're going to be collecting these samples out at police substations and at the police training academy. And then when patients become symptomatic, we're going to be doing some of the visits at their homes because they will be in quarantine and it will be the safest place for us to collect those samples. The other thing about the study from a logistics standpoint is having the right type of research staff to collect these samples. And nasopharyngeal samples are a more invasive procedure. And so we actually have been hiring paramedics who are licensed and able to collect these samples as part of our research team. The next slide, Ann, please.
Trying. Sorry, something happened.
That's all right. I needed to.
There we go. Okay.
So as I mentioned, we're going to be looking at them for every 6 months. We're going to follow those first responders and household contacts. Initially, we're only going to be looking at adults. However, as these adults in the households become symptomatic, we do have an option to assent children to collect saliva samples. So we won't subject them to blood or nasopharyngeal sampling, but because we don't understand all of the transmission that might be involved in the household, we are going to look at the children potentially if they consent as well. And we'll closely monitor. And then all of the samples that we're collecting clinically are going to be used for clinical tests and are going to help look at some of the basic science and bench science research to look at the antibody responses to COVID with no infection, with presence of infection, and recovery from infection, and again with vaccination.
Okay, now we have project 2.
Yeah, I'll chime in here. So project 2 is an exciting area because there's some of the more critical unanswered questions that if we really want to mitigate COVID-19 and transmission and disease, we're going to have to address them. So as an example, what underlying host and viral factors determine drastically different outcomes from SARS infection? Or how about, does natural infection confer immunity to reinfection? So these are really tough questions and they're not easy to answer, but Project 2 is going to try to address these. And one of the exciting parts of how these are going to be addressed is really by evaluating the serologic and molecular determinants of COVID-19 severity and immune protection. Now, one of the exciting questions within this is how do we address whether antibodies to seasonal endemic coronavirus or common cold coronavirus can provide some protection from that severe disease? Or can it make things worse through some sort of antibody-dependent enhancement? So we don't know the answer to these questions, but the exciting part is that we have a team who can evaluate this. To address this, Anastasia Vlasova and Linda Safe are developing assays that will distinguish antibodies specific to SARS-CoV-2 or the common cold coronavirus antigens, or those that cross-react with both. And you can see these in the lower left-hand corner. Now, as an example to kind of show you what you're able to do here, that you can actually measure these, is look on the far right-hand side. Here you see SARS-CoV-2 convalescent serum, either from people in blue or non-human primates, which you have in red. And you can see in the first S2-1 where the N peptides, the CoV-2 epitopes from the N peptide are actually bound to either the blue and the red, which is again the people or the non-human primates. However, the normal human serum in gray does not. Now, instead, both types of these human serum will bind the common cold coronavirus, whereas the NPH will not. And so this is one example of actually using this assay. Give me a click, Ann, if would you mind? Now, another example of this, just a little click.
Ah!
No, it's okay. Okay, there we go. Another example of this is we can actually assess this in our severe COVID patients, and you can see high levels of antibody against that N protein epitope from SARS-CoV-2, again, with little or no cross-reactivity with uninfected patients. So we're able to do these. So with these assays in hand, we can now assess the levels of common cold coronavirus in our large cohorts and follow outcomes, not just if individuals become infected, but also how pre-existing common cold coronavirus B cells and their antibodies might impact immune response to a vaccine. So in the end, this is opening up a whole new area of investigation where we can look at these cross-reactivities and better understand what the next steps are. I believe this might also be me, isn't it?
Yeah, it's you.
Okay, no problem. So finally, Dan Jones is also working on this, and he has a swab for transcriptomic assays for expression of key host and viral genes, as well as their genetic variants. We will use all of this data to monitor innate and adaptive immune response to genes in infected individuals, as well as the co-infections by other respiratory viruses. Now, this data itself will probe the genetic variants in the host genes. And I know we've been hearing a lot about genetic variants and things like that, but these are important, since we'll be able to further look at viral receptors and inflammasome components. So it becomes very important for us to continue monitoring these genetic variants as the virus itself, both in primary and secondary infections, or even common chronic infections, will continue going through our population. Now, keep in mind, this may also occur in our vaccinated individuals. So lots of questions, lots of changes which could occur, and that we're going to be able to follow using these techniques from Dan Jones's team. Go ahead, Ann.
So then project 3 is really looking at the response to changing serological and viral information around COVID-19. My co-PI for this project is Cindy Seek, who is an expert in health behavior and health promotion and health education. And so we have designed this project to look at improving our understanding of communication around the efforts for COVID testing, need for testing, implications, need for follow-up, and then regularly update the findings from projects 1 and 2 in the SARANET collaboration into actionable guidance and using our Stop COVID website. So this is really the project that tries to pay attention to what's going on across the projects and across the SARANET to try to figure out if there's new questions we should be asking of particular populations, if there's new angles that we should be considering for our communications, and so forth. The interesting thing about this particular project is that we have, just as with the serologic project, project 2, this is beyond first responders. So we're looking at first responders and their household contacts, but we're also looking at at-risk patients. We're looking at cancer patients under active treatment. We're also looking at community members that are members of state agencies, and we're looking at providers. So having that expanded view and snapshot over time is part of the advantage of this particular project. So aim 1 is really to improve our knowledge of how organizations and individuals understand and communicate the concepts, opportunities, and implications of viral and serological testing, and I would say now as well as vaccines. So as I said, our key informants are the people that we're interviewing and surveying over time, have gone beyond the first responders. We have embedded some questions in our enrollment survey that Jen and Ash are working on with our first responder populations to look at certain ideas around things like symptom control. We've reached out to various members of the OSU community and have gotten really interesting questions from Jan Keithel-Glazer around stress and immune response, depression, and so forth. And then from Sai Marai in ophthalmology, we've gotten questions about eye symptoms. So we've been able to embed those questions into our surveys, and we'll be able to follow those over time. So what we're looking at in our interviews and our surveys are the perspectives of these different populations about testing, barriers, interpretation, need for follow-up. And what we found, even just in our pilot interviews, is a considerable amount of confusion about the guidance. And so what we're trying to figure out is, with this changing information over time, and I would say that this information is still changing daily, what can we do about this, and how can we direct people to the right place? The second aim of this project is looking at, as new information becomes available from projects 1 and 2 in CERANET, how do we figure out best practices related to the communication and new findings? How do we look at kind of trusted sources of information and so forth? So looking at this, what do we do and what do we present to the different audiences in different ways? And things like, you know, how do you get the survey questions to the right reading level for the right population? It might be a different survey question for a provider population versus just the general population that is served by the Columbus Department of Health, so Public Health. And then the third aim is this aim around guidance. So how do you provide guidance for organizations? So we're hoping to become one of these trusted providers of information, and provide communication tools that include this guidance, information about risks, how to deliver messages about test results and implications, reducing barriers. Share this with the CERANET. There's I think 1 or 2 other projects across the CERANET that have a focus something like this. And so we're going to be in contact with them as they advance their science as well. And this is kind of a mock-up of the website that we put into the application. It doesn't actually look like this. That's just way too many words, but we will be making that guidance available on the website. And I'm going to turn it over to Ash to wrap up and
Perfect.
go for it.
Yeah, so thanks everybody for bearing with us as we've kind of given you a 20,000 foot view of the overall project. Of course, this has been a labor of love, and amazingly enough, all of us have become very fast friends through this whole process of putting all this together. Now, the key thing though is the science. So the project will address a number of critical questions that I think will really change the dynamics of how we look at SARS-CoV-2 infection and disease. So first, we will understand the prevalence of SARS-CoV-2 infection and disease severity among high-risk exposure groups, along with the impact of their PPE use and transmission to household contacts. Now, look at this concept itself. This topic itself becomes one of the most critical concepts that we need to address. We don't even know how long we should hang on to people, and we don't have data to say how long you should really keep somebody out of work, or what do you do with somebody who is an asymptomatic carrier, or is it important for identifying an asymptomatic carrier? All these things impact a lot of our decision making, even on the front lines of who do we put in the field. So these fundamental questions about infectivity and transmission are critical for us to have a better understanding about how to mitigate the disease process. Second, we're going to get a better understanding of whether positive PCR results consistently equate with infectious virus, right? And we're going to need the facilities, and we do have them, like BSL-3 assays and places to do these assays, which become critically important to, again, further understand this. We will look at the evolution and longevity of antibody response to natural infection and reinfection over a large range of symptoms. Because we have a pretty large population of individuals we'll be looking at, we're going to be able to look at not only their comorbidities and what that looks like, but also everyone from mild, moderate to severe infections, and seeing where all their response happens. Now, when we start thinking about antibody response and all these kind of concepts, I'm going to come back to the concept of viral protection in a second, but that ties into this area. Next, we'll evaluate the links between viral load, host viral genetics, and common cold coronavirus exposure to disease and immune responses. This is a lot of what I talked about when we talked about Project 2 and how we look at those links between the disease processes. Now, finally, I think one of the most important concepts and one of the biggest pivots we've had to do is concerning the post-vaccination state. We will examine the humoral immune responses in naive and previously infected or reinfected individuals post-vaccine. And we'll look at reinfection or infection post-vaccination. So we'll be able to really look at those viral genetics for our population. Now, these pieces we had already built into the grant of the post-vaccine age on the off chance that the vaccine gets released. So all of these different goals and deliverables are put together a larger package which links all three of the specific aims even though we come from all diverse populations and diverse skills and expertise, we still have this cogent theme throughout the whole application, which in the end will give us a deliverable where we can really move the needle forward when it comes to managing COVID-19. So at this point, I believe we're at the point where we can start talking a little bit about any questions or comments that you guys might have, or any guidance about how to go through these experiences. I definitely have my little laundry list of the fun things that I thought were exciting about this whole process, but I do want to open it up to the group first. Thank you, guys.
Thank you for your presentation. So I want to urge anyone who has any questions to submit it using the question and answer function at the bottom of Zoom. And I'll proceed to facilitate that discussion. But I'd like to start off with a question, which is, can you speak to how you deal with the language issues that challenges interdisciplinary research, given your very different expertises and disciplines in creating the proposal in such a short period of time?
I think we all have comments on this. I'll get it started, then I'm going to kick it back to Ann and Jen. So I think the language aspect of what you just referred to, Tim, is so critically important. We had a whole bunch of different languages here, right? Like from the pre-hospital space, both police and fire are slightly different. And so you have very different languages there. And then of course, clinical emergency medicine and just general EMS care and how you manage that. Of course, infectious disease and how you manage surveillance and prevalence and how to evaluate that, not to mention how you do true transmission type statistical evaluation, right? In a large population. How do you follow people? How do you look at that question? And that's just looking at population dynamics. We're not even getting into the science yet. Right. So when you put Linda and Jean in a room and you start talking about all of the common cold coronaviruses and all the epitopes and things like that, and Sean Liu then walks in and all your head starts spinning because of all the different terms which are there, which make absolutely no sense to me. Well, a little better now, but it's still unbelievably challenging. Ann, your thoughts?
Yes, I would echo that. Even though I was actually trained in the basic sciences, it has been quite some time, and it has been a fire hose of learning. I will say, having heard this presentation now probably 4 or 5 times, I think, gosh, you finally got it through for project 2 there. I was following along with project 1 just because of the logistics that Jen has been managing so beautifully. But I think the other side is that each of the vocabularies, as Tim pointed out, is completely different. Even though I'm a health services researcher, I didn't actually know that pre-hospital was a thing. So to think about that, in addition to kind of how do we work with the science and how do we feed things back, has been really, really an interesting challenge. It is, as Ash said, some of the conversations are just like whoosh, 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 and I gotta say that these slides with these test tubes and things is just completely different from my prior experience. But I would say that with this amazing team, as we move forward and as we are really getting going on this project, it is extremely exciting, and I am delighted to be a part of it. When Jean first called, I thought he was crazy. Like, how in the world could I contribute to this type of a project? And Jean's like, well, Peter Moeller said, to give you a call. I'm like, okay, I guess we can figure this out. And kind of listening to what was going on and trying to pivot in my own head about kind of the P30 that was winding down, which was similarly complicated, but had similar science. It wasn't really the basic sciences that we had in the P30. So to kind of pivot to think about, okay, this is a big organizational question. Okay, I can do that. And now let's think about how to embed the science. And oh, by the way, I have these collaborators that I can work with, and they will answer these questions. So I didn't feel alone in terms of trying to figure that out. This group has been so collaborative that, we have a question, you text it. I would say that we're getting up to speed on the technologies that we're using. We've mostly mastered Zoom, but even when the score came out, Jean wasn't. I texted Jean this score and said, "Hey, are you sitting down? This is like the best score ever." And it was actually, it was the first center that was funded, so it was pretty impressive. But each of us is, kind of has our strengths and our non-strengths in terms of where we're going. And I think that's the beauty of putting this all together with the different things. And I think Jen probably has a lot of things to add about kind of her clinical research experience and trying to train
train people about things like protocols and
And people not being mice. So I totally echo what Dr. Panchal and Dr. McInerney said. I think the biggest thing is a lot of us have had our feet in multiple worlds and there's a lot of overlap. And so when people were talking about some of the basic science, I was able to help translate to other colleagues and vice versa. And so I think we've each taken our, okay, I don't understand this, and had the side conversations if we couldn't have it in a big conversation. But the other thing that really came in to light for me, and I was very dreary-eyed as we wrote the proposal, because I was doing enrollments at 6 o'clock in the morning, and then we were having 2 o'clock meetings, was all the other infrastructure that supports us, the grants management, the finance, the OSP, the IRBs, the red cap, you know, accounts receivable and logistics and supply chain. This whole project wouldn't be possible without all of that infrastructure that we have at OSU. And I think that we are very blessed and fortunate to have all that at this institution, because as I talk to my colleagues, other places around the country, it's just not there.
Yeah, one last thought, just for all of us, for just our own little deliverable, so we can walk away from this conversation with something that really kind of moves us forward. So one of the fundamental things that I always experience on Zoom calls is people talking over each other, right? We did all of this over Zoom. And so one of the things that now in hindsight, as I look back to those early conversations, was the humility that people would have to sit on the sidelines and just listen when someone was talking, and to really internalize what they were saying. And the bigger thing for me was to hear someone like Linda, for instance, who is world famous, internationally known, to be able to, who actually steps in and says, I have absolutely no clue what you're talking about right now. And that's okay. And all of us did that. But it really sets the tone for really walking into these kind of collaborative experiences, really with your hat in hand, and really be open that everyone's going to bring something to the table. It's all going to be radically different from what you do. And so the fact that someone does not understand what you're talking about at this moment does not in any way discount their opinion. And so those are real challenges, because you're leaving ego somewhere else, right? When you're coming and sitting down with this group of individuals, and all of us were powerful in our own little spaces, but we don't necessarily know what each other's reputations are. And so just a little deliverable, I think, for me, when I walked away from this experience, was that type of role modeling that happened top down, which made a huge difference.
In follow-up to your comments, one of the questions I have is how often, what I see a lot in especially amongst clinical researchers, is trying to do research off the side of your desk. And writing a large grant off the side of your desk seems to be a Herculean task. How often, getting very pragmatic, how often did you meet? And how often were you talking? I think one of the things that is a challenge is that people think that it's just about the idea, and that you can just write things and you'll get and if you don't get funded, it's because the reviewer didn't understand. You know, how much time did you actually spend talking to one another? And what does that accelerated collaborative process look like in very pragmatic terms?
It was a full-time job, I think, for that month of July. There was constantly conversation going on. And I think that one of the things that made this grant score so highly is that we were able to write it so that it was understandable to every reviewer. As a grant reviewer in my professional organization, I'm constantly telling people you have to be able to sell your niche to somebody else. And because there was no one niche for this project, we all had to say, this doesn't make sense, it doesn't make sense, and bring it into some of that more general scientific layman term, for lack of better saying. So my phone battery probably needed to be replaced in July, and I'm sure the same was for Jean and Ann and Ash and everybody else who was writing on the team.
Yeah, I think to Jen's point, there were a lot of meetings. I mean, honestly, I think we probably met every other day, if there wasn't conversation every day. I think for those of us most heavily involved, there was definitely conversation every day. It's just whether or not we scheduled a Zoom call with the broader group. The other thing that happened is that, everybody, you'd have your meeting and then you would have your homework. And the thing about this group was that people actually did their homework. So, Ash was like, okay, I have this contact at Delaware County, I'll find this person. And he did. And then the email came back and then you had that information to work with. And I think just another logistical twist, which I thought was entertaining, is that to get the application together at the end, you also had to come to agreement about what was going to be the font and what was going to be the spacing and how many pictures were you going to have and things like that. So, Linda has done grants in certain ways and Ash has done grants in certain ways and I've done grants in certain ways. That all came together and it was, Gene as the contact PI was the guy that put it all together and made sure it all looked good. We had plenty of people, many extra sets of eyes, but trying to figure out that the story was cohesive and that kind of thing. And for him to step up and do that. As Tim knows, I'm a total micromanager of my own grants. So there was definitely extra eyes, but I think that commitment to make it as good as humanly possible was huge and was collective.
Yeah, I think just going to piggyback on what you were saying, we had project teams, right? So we had project 1, project 2, and project 3 teams. And then we had the overall section that we had to write too. So each of the major sections had its own things that had to be written. Then we had the overall to write. The MPIs focused on the overall to write together, and we divided that labor and we had small focus groups which would get together. So project 1, me, Dan, Jen, like we would all get together and talk about our own projects. So we had these sub-project meetings. In the face of these larger projects, we bring that product back to the main team for them to beat it up. Let's be honest, we needed that, right? And so it was that collaborative interplay that you're having. So yes, you'd have a lot of meetings. Obviously, I'm very clinically active, both in the pre-hospital space with my agencies as well as the emergency department. I put a lot, other than doing my shifts, all the rest of my research, everything was on hold. This was life at the time. But I will be honest, I had a blast. Like, this was exciting. This was fun. It was inspiring. And that made the lift that much easier. That's probably good for now.
But I think that's actually huge because it felt like a positive thing in the context of COVID, which at that point was so negative and so depressing. To think that we had the guts to kind of pull this all together in a way that was truly exciting scientifically and from a research standpoint across the board. I think that was really impressive. The other thing is, to Ash's point, in addition to those project team meetings that were happening in between the big group meetings, in addition to the sections of the grant, we also had the cores. So we had the core leads writing their sections that then had to be embedded, and then this whole kind of making it interwoven. So we had the overall, the 3 cores, the 3 projects, and then across the grant, we wanted to make sure that we reference each other. So in project 3, we're going to use the findings from project 2 and core A and blah, 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 blah. So to make it seem like we had our act together, which we do, of course, but that was kind of part of it too.
And I think the other thing that made it all come together for solidifying that message was the visuals. I think Dr. McInerney and Dr. Panchel had drawn out some stuff, and then Alice had pulled in people to come up with several of the other graphics. And so when those graphics started coming together, I think everybody started seeing the total picture. And I think that also helped that final solidification.
One last thought, I'm going to let us go to the next question. So for any of you who are just starting on the grants process, because even though I was an MPI on this, I still feel like I'm very young in the grants process personally, but I cannot highlight how important it is to have key support people who understand how to put together budgets and things like that in the background. Alice and Jen floated our boat because we were all able to just sit around and talk about science and geek out. Whereas the nuts and bolts of what needs to actually get done, someone else was really focusing on 100% of their time. So none of this could ever happen without Alice and Jen. I mean, they were the ones who made sure that this ship would actually sail. It's great to have ideas, but if you cannot enact those ideas, you can't move forward. So make sure that as you start thinking about a grant in any way, shape, or form, put together the team, not just the scientists. Think about who's going to be the closers for you, because we were not the closers. The closers were Alice and Jen.
So I'm looking on the Q&A and don't see any right now. I know we have a break that immediately follows this. Becky, did you want did you have any questions you wanted to ask or raise? And if not, I can move us to the break.
I think we can move to the break.
Wonderful. Thank you all for coming and presenting today.
Actually, I do want to say one word. I think that hearing your insight, the ways to do really collaborative team science, that respect for different perspectives, backgrounds, really embodies what we're talking about, really extending to that next level as we talk about DEI. And so, you know, it begins to be somewhat of a blueprint for us to think about how do we really make a difference. And I hope that we really capture these types of things. I'd encourage your group as well to think about writing papers on this, of how do you actually move forward this. And it may sound silly, but in fact, these are what all of us, whether we're in the early part of our career or like me, at the latter stages of it, that there are so many things that one have to traverse. It would be nice if we all had 6 months, whether it be a pandemic or an RFA. We always have tight timelines. And so understanding how do we bring together people and how do we do it in a respectful fashion? How do we disagree respectfully? I think would be an incredible sharing of lessons learned. So thank you so much for doing this today.
Thank you. It's been a great opportunity. We appreciate it.
Wonderful seeing you all again, and we'll leave to a break and we'll see each other. I think we start again with our next presentation at 11:15, which is rapid COVID-19 diagnostic testing engineering response to COVID-19. Thanks, everybody.
Thank you.
Thank you.
Thank you.